question, we're going to be looking at the Edexcel Combined Biology Paper 1, and this is from the year 2018. Please make sure that you've got a pen, some pencils, a ruler and a calculator. In the first question, we're told obesity increases the risk of a person developing cardiovascular disease. Losing weight can reduce the risk of disease occurring. Explain why exercise can cause weight loss. Well, exercise requires energy, and that energy will come from the stored fat in the body. For part B, we're shown figure 1, which shows a gastric band fitted to a stomach. Explain how a gastric band helps a person to lose weight. So the gastric band will reduce the volume of the stomach, which will then restrict the amount of food that's taken in. In part C, we're told that BMI and waist-hip ratio can be used to find out if a person is obese. Figure 2 shows some data of two males. So we have male A with a BMI of 27.3 and a waist-hip ratio of 0.85. And then we have male B. We don't know the BMI, but we know that the waist-hip ratio is 0.81. And we're told that male B has a mass of 72 kilograms and a height of 1.81 meters. We've got to use the equation to calculate the BMI of male B. So the BMI is going to be the mass, which is 72, divided by the height squared, so 1.81 squared. And if we calculate that out, we get an answer of 21.977, and because we've got to give our answer to three significant figures, our answer would be 22.0 to three significant figures. Now, in this part of the question, we have figure 3, which shows whether the BMI indicates if a person is underweight, normal, overweight, or obese. Males with a waist-hip ratio above 0.9 are defined as abdominally obese. Explain what the BMI and waist-hip ratio for a male A shows about his waist distribution. So what I've done here is I've taken the information and put it in so we can remind ourselves of what it was for male A. He had a BMI of 27.3, so that shows that although he's overweight, he's not obese, and his waist-hip ratio was 0.85, so it's not above 0.9, which means his weight must be more evenly distributed. It's not just around his waist. In question 2, we're given figure 4, which shows two potato chips. We've got the length of X, the length of Y, and the length of Z as labelled in figure 4. And we've got that for A and B. Then we've got the total surface area of the four sides, which has been worked out for A but not for B. The total surface area of the top and bottom in centimetres squared, and then the total surface area of the chip in centimetres squared. And our question is, calculate the total surface area of potato chip B using the formula total surface area is 2x times y plus 2x times z plus 2y times z. So if we put our numbers in, we've got 2x, which is 5, times y, which is 2, plus 2 times x, which is 5, times z, which is 2, plus 2 times y, which is 2, times z, which is 2. And if we calculate all of that out, we get an answer of 48 centimetres squared. So in this part of the question, we're given figure 6, which for potato chips A and B gives us the increase in mass in grams. So so the potato chips were placed in distilled water for 20 minutes. Figure 6 shows the increase of mass. So A increased by 0.1 grams and B increased by 0.3 grams. So we've got to explain why chip B has increased in mass more than chip A. Well, if we remember, or if we look back to the table, chip A has a much smaller surface area compared to chip B. Therefore, it's chip B is going to be able to absorb more water. Then for the next part of the question, part 3, it says potato chip A is transferred from the distilled water into a concentrated salt solution. Explain what will happen to the cells in potato chip A. Okay, so now we've put it into a solution which is concentrated salt solution, so there's less water outside. So the cells are going to lose water by osmosis from an area of high concentration, which is in the cells, to an area of low concentration, which is outside the cells, through the partially permeable membrane. So now we're told the potatoes of a potato plant develop underground. Explain one difference in the subcellular structure in a cell in the potato and those found in the leaf of the potato plant. Because the potato is underground, it does not need any chloroplast for photosynthesis. From September 2013 to March 2014, there were 2,800 and from September 2013 to March 2014, there were 2,831 cases of scarlet fever in the UK. From September 2014 to March 2015, there were 5,943 cases of scarlet fever. Calculate the percentage increase in the number of cases of scarlet fever between the periods of September 2014 to March 2015 and September 2013 to March 2014. So first of all, we need to know from March 2014, what was the increase in number? So if we do 5943, take away 2830, that therefore tells us that there was an increase of 3,113 cases. Now, we had an increase of 3,113 cases from 2,830. So to turn that then into a percentage, we would times by 100. And if we calculate that out, that gives us a percentage increase of 110%. In this part of the question, we're shown figure 7, which is actually showing us a microscopical picture of the bacteria. It says some bacteria are motile, meaning they can move themselves. Why is Streptococcus bacterium not motile? And the correct answer there is going to be because it does not have flagella. Flagella is like the tail which helps it move around. Part B says patients with scarlet fever can be treated with antibiotics. New antibiotics need to be tested before they can be used in patients. Which is the correct sequence for the development of a new medicine? So the correct procedure should be it's tested on cultured cells, then it's tested on healthy volunteers, and then we have a double-blind trial. So instantly we can see it's going to be between B and D. And the correct one out of those two would be D because it's tested on the healthy volunteers before the double-blind trials. Most cases of scarlet fever occur in children. Adults have usually developed immunity to a toxin that the Streptococcus bacteria produces during infection. Explain how an adult develops immunity to the toxin. So, first of all, when an adult has been exposed to the pathogen, and this could be through immunization or vaccination, the body is stimulated as an immune response system. And what happens there is that the body produces antibodies that would fight off the disease. But the body also produces memory lymphocytes, and the memory lymphocytes will remember the specific antigen. So if the pathogen enters the body again, the antibodies are produced in bigger numbers and much more quickly. In 2012, two scientists were awarded the Nobel Prize for their research on stem cells. They showed that adult cells could be reprogrammed to become cells with the properties of embryonic stem cells. Describe the possible benefits of this research, and it's a 3-mark question. So, the first thing that we can say is that stem cells have the potential to produce any cell type. And because we can reprogram the adult cells to become cells with properties of the embryonic stem cells, that means that we've got no need to use embryonic stem cells, and there's going to be less of a chance of a patient rejecting their own cell when we're using their own cells for stem cells. You could also say that the stem cells could be used to treat diseases that we can't treat at the moment, and also can be used for things like transplants. For this next question, we need to look at the stages of mitosis. So we've got different parts, P, Q, R, and S, and we've got to put them in the correct order. So to start off with, what I would do is I would just number them in what I think is the correct order. So this seems to be the correct order. And then if we look at our options, we're looking for R, Q, S, and P. So R, Q, S, and P. So the correct answer is B. Then for part 2, it says the stages of mitosis labelled in S in figure 8. So we're looking at this stage here. What is the correct labelling for that stage there? And that would be anaphase. And then for part 3, it says interphase is part of the cell cycle. Describe what happens in interphase. So during interphase, before a cell can divide, it first has to grow or increase the amount of subcellular structures, such as the mitochondria and the ribosomes. It then duplicates, so there's one copy of each new cell. So for part C, figure 9 shows a root tip with cells in different stages of mitosis. This image has been magnified 400 times. Explain how a magnification of 400 times can be obtained using the lenses on a light microscope. So basically what we've got to do is we've got to think of our two lenses, which is the objective lens and the eyepiece lens, and we've got to get them to be multiplied together to equal 400. So the objective lens is usually the most magnification. So we could use an objective lens of 400 times and combine it with an eyepiece lens of 10 times. Potato cells contain the enzyme catalase. The enzyme catalyzes the breakdown of hydrogen peroxide into oxygen and water. So figure 10 shows what happens when a student placed a potato disc in a 5% hydrogen peroxide solution. So we've got our potato disc at the bottom, and then we had our hydrogen peroxide solution. And over time, oxygen bubbles will cause the potato disc to rise. The student measured the time taken for the potato disc to rise, and then repeated the investigation using 10, 15, and 20% concentrations of the hydrogen peroxide solution. Which term describes the hydrogen peroxide in this reaction? So the hydrogen peroxide would be the substrate. Then for part 2, we're told the potato discs all had the same mass. Explain why the student used the potato discs with the same mass. So we're using the same mass because it's a control variable and we need the mass to be controlled so we can compare the different results. So in part 3, we're asked to state 2 other factors that need to be kept the same to improve this investigation. So we could have any 2 from things like the temperature has to be kept the same, the volume of hydrogen peroxide must be kept the same, the pH, the size of the test tube, the type of potato, the surface area of the potato. So any 2 from those answers would be ideal here. Figure 11 shows the results of the investigation. The student calculated the rate of reaction using 1 divided by the time taken in seconds. So we have the concentration of hydrogen peroxide solution, we have the time taken for the disc to rise, and then we have the rate calculated using the equation. And the question is, state and explain a conclusion based on these results. So we can clearly see that as the concentration of hydrogen peroxide solution increases, the rate of reaction increases. And this would be due to, as we have a higher concentration of hydrogen peroxide, that means there's more substrate. And if there's more substrate, then we're going to increase the number of collisions. And if we increase the number of collisions, that means that more oxygen is going to be released and the reaction would be faster. The student repeated the investigation with a 25% hydrogen peroxide solution and recorded a time of 75 seconds. Calculate the rate of reaction for the 25% hydrogen peroxide. So the rate of reaction is 1 divided by the time taken. So that would be 1 divided by 75, and that gives us an answer of 0.013333, and giving our answer to the correct number of decimal places, that would give us an answer of 0.013. Then for the next part of the question, we are told that the student decided that the rate for 25% hydrogen peroxide solution was not anomalous. Give a reason why the result was not anomalous. Well, the result cannot be anomalous because the substrate, the hydrogen peroxide, is not the rate limiting factor, so it wouldn't be anomalous. So in question 6, part A, yeast cells can be genetically modified to produce a painkiller. The painkiller is usually obtained from opium poppies. One method for genetically modifying a yeast cell is using a plasmid containing the desired gene. Explain how a gene can be inserted into a plasmid, and that's 2 marks. So we can use a restriction enzyme first of all to cut open the plasmid, and then we use the ligase enzyme to insert the gene. Then for the next part of the question, we are asked to discuss the possible benefits and risks of producing painkillers from genetically modified yeast cells rather than extracting the painkillers from poppies. So in this question, we have figure 12, which shows the structure of a DNA nucleotide. So here we have the base, here we have the sugar, and here we have the phosphate. And our question is, structure A is so if we look at structure A is the sugar. For this last question, we're told that in 2003, the first complete human genome was sequenced. The genomes of different people have different small changes in the sequence of the DNA bases. Describe how these changes in DNA sequence can affect the individual and how sequencing a person's genome could influence their medical treatments. So first of all, DNA is made up of 4 different bases, and when we have changes to the base sequence of the DNA, this leads to mutations. And the mutations will affect the person's phenotype, so will lead to variation. Now, when we DNA sequence an individual's genome, what that can do is it can help us to identify if there are any genetic disorders or the likelihood of a particular person developing a particular genetic disorder. And this in turn can allow the individual, if they know they're at a higher risk of developing a particular disorder or a particular disease, it could lead to them maybe changing their lifestyle to try and reduce the possibility of that happening. And then lastly, we need to think about the impact on medical treatment. So when we're looking at sequencing a person's genome, then we could understand whether a particular person would be responsive to a particular disease, and then we could try and look at providing tailor-made medical treatments to that particular person to ensure that the medication is more effective. We would also have a better understanding on preventative medicines for a particular person. So if you've managed to keep up and gone through the whole paper with me, well done. Doing as many of these past papers will help you to get those higher marks in the assessments that you have coming up. I do strongly recommend that you subscribe to the channel. What this will do, it will give you easy access to all the new content that's being added on a frequent basis, not just past papers but topic learning as well.